Hello. Topic of our lectures is physiological properties of the heart. The human heart is located within the thoracic cavity, medially between the lungs in the space known as the mediastinum. Figure shows the position of the heart within the thoracic cavity. Within the mediastinum, the heart is separated from the other mediastinal structures by the talc membrane known as the pericardium or pericardial sac, and seat its own space called in pericardial cavity. Membranes, surface features, and layers of the heart. Membranes. The membrane that directly surrounds the heart and defines the pericardial cavity is called the pericardium or pericardial sac. It also surrounds the roots of the major vessels or the areas of closest proximity to the heart. The pericardium, which laterally translates and around the heart, consists of two distinct sublayers: the sturdy outer fibrous pericardium and the inner serous pericardium. The fibrous pericardium is made of talc, dense connective tissue that protects the heart and maintains its position of the thorax. The more delicate serous pericardium consists of two layers: the parietal pericardium, which is fused to the fibrous pericardium, and an inner visceral pericardium, or epicardium, which is fused to the heart and is part of the heart wall. The pericardial cavity, filled with lubricating serous fluid, lies between the epicardium and the pericardium. In most organs within the body, visceral serous membranes, such as the epicardium, are microscopic. However, in the case of the heart, is not a microscopic layer, but rather a macroscopic layer, consisting of a simple squamous epithelium called a mesothelium, reinforced with loose, irregular or areolar connective tissue that's attached to the pericardium. This mesothelium secretes the lubricating serous fluid that fills the pericardial cavity and reduces friction as the heart contracts. Layers. The wall is composed of three layers of unequal thickness, from superficial to deep. These are the epicardium, myocardium, and endocardium. The outmost layer of the wall of the heart is also the innermost layer of the pericardium. The epicardium or the visceral pericardium discussed earlier. The innermost layer of the heart wall, the endocardium, is joined to the myocardium with a thin layer of connective tissue. The endocardium lines the chamber where the blood circulates and covers the heart valves. It's made of simple squamous epithelium called endothelium, which is continued with the endothelial lining of the blood vessels. Once regarded as a simple lining layer, recent evidence indicates that the endothelium of the endocardium and the coronary capillaries may play active roles in regulating the contraction of the muscles within myocardium. The endothelium may also regulate the growth pattern of the cardiac muscle cells throughout life, and the endothelials, its secrets create an environment in the surrounding tissue fluids that regulates ionic concentration and state of contractility. Endothelins are potent vasoconstrictors and in a normal individual establish a homeostatic balance with other vasoconstrictors and vasodilators. The middle and thickest layer is a myocardium made largely of cardiac muscle cells. It's built upon a framework of collagen fibers plus the blood vessels that supply the myocardium and the nerve fibers that help regulate the heart. It's the contraction of the myocardium that pumps blood through the heart and into the major arteries. The muscle patterns is elegant and complex as the muscle cells swirl and spiral around the chambers of the heart. They form a figure-8 pattern around the atria and around the bases of the great vessels. Deep ventricular muscles also form a figure-8 around the two ventricles and proceed towards the apex. More superficial layers of ventricular muscle wrap around both ventricles. This complex swirling pattern allows the heart to pump blood more effectively than a simple linear pattern would. Also, the ventricle on the right and left sides pump the same amount of blood per contraction. The muscle of the left ventricle is much thicker and better developed than that of the right ventricle. In order to overcome the higher resistance required to pump blood into the long systemic circuits, the left ventricle must generate a great amount of pressure. The right ventricle does not need to generate as much pressure, since the pulmonary circuit is short and provides less resistance. Internal structure of the heart. Septa of the heart. The word septum is derived from the Latin for something that encloses. In this case, refers to a wall of partition that divides the heart into chambers. The septa are physical extension of the myocardium lined with endocardium. Located between the two atria is the interatrial septum. Normally, in an adult heart, the interatrial septa bears an oval-shaped depression known as the fossa ovalis, a remnant of an opening in the fetal heart known as the foramen ovale. The foramen ovale allowed blood in the fetal to pass directly from the right atrium to the left atrium, allowing some blood to bypass the pulmonary circuit. Within, second after birth, a flap of tissue known as the septum primum that previously acted as a valve closes the foramen ovale and establish the typical cardiac circulation pattern. Between the two ventricles is a second septum, known as interventricular septum. Unlike interatrial septum, the interventricular septum is normally intact after its formation during fetal development. It's substantially thicker than the interatrial septum, since the ventricles generate far greater pressure when they contract. The septum between the atria and ventricle is known as atrioventricular septum. It's marked by the presence of four openings that allows blood to move from the atria into the ventricles and from the ventricles into the pulmonary trunk and aorta. Located in each of these openings between the atria and ventricle is a valve, a specialized structure that ensures one-way flow of blood. The valves between the atria and ventricles are known generally as atrioventricular valves. The valves at the openings that lead to the pulmonary trunk and aorta are known generally as semilunar valve. In this figure, the atrioventricular septum has been removed to better show the bicuspid and tricuspid valves. The interatrial septum is not visible since its location is covered by the aorta and pulmonary tract. Since these openings and valves structurally weaken and atrioventricular septum, the remaining tissue is heavily reinforced with dense connective tissue called the cardiac skeleton or skeleton of the heart. It includes four rings that surround of openings between the atria and ventricles, and the opening in the pulmonary trunk and aorta, and serve as a point of attachment for the heart valves. The cardiac skeleton also provides an important boundary in the heart electrical conduction system. Chambers of the heart. Figures show different chambers of the heart. There are present right atrium, right ventricle, and left ventricle. Right atrium. The right atrium serves as receiving chamber for blood returning to the heart from the systemic circulation. The two major systemic veins, the superior and inferior vena cava, and the large coronary veins, called the coronary sinus, that drain the heart myocardium empty into the right atrium. The superior vena cava drains blood from regions superior to the diaphragm, the head, neck, upper limbs, and thoracic region, and empty into the superior and posterior portions of the right atrium. The inferior vena cava drains blood from areas inferior to the diaphragm, the lower limbs and abdominopelvic region of the body. It too empties into the posterior portion of the atria, but inferior to the opening of the superior vena cava, immediately superior and slightly medial to the opening of the inferior vena cava on the posterior surface. The atrium is the opening of the coronary sinus. These thin-walled vessels drains most of these coronary veins that retain systemic blood from the heart, while the bulk of the internal surface of the right atrium is smooth. The depression of the fossa ovalis is medial, and the anterior surface demonstrates prominent ridge of muscles called of pectinate muscles. The right auricles also has pectinate muscles. The left atrium does not have pectinate muscles except its auricle. The atria receive venous blood on a nearly continuous basis, preventing venous flow from stopping while the ventricles are contracting. While most ventricular filling occurs while the atria are relaxed, they do demonstrate a contractile phase and active pump blood into the ventricles just prior to ventricular contraction. The opening between the atrium and ventricle is guarded by the tricuspid valve. Right ventricles. On figure, you can see blue colors ventricles. The right ventricles receive blood from the right atrium through the tricuspid valve. Each flap of the valve is attached to strong strands of connective tissue. The chorda tendinea, literally tendinous cords, or sometimes more poetically referred to as heartstrings. There are several cord tendinea associated with each of the flaps. They are composed of approximately 80% collagen fibers, with the remainder consisting of elastic fibers and endothelium. They connect each of the flaps to a papillary muscle that extends from the inferior ventricular surface. There are three papillary muscles in the right ventricles called the anterior, posterior, and septal muscles, which corresponds to the three sections of the valves. When the myocardium of the ventricles contracts, pressure within the ventricular chamber rises. Blood, like any fluid, flows from higher pressure to lower pressure areas, in this case towards the pulmonary trunk and the atrium. To prevent any potential backflow, the papillary muscles also contract, generating tension on the chorda tendinea. This prevents the flaps of the valves from being forced into the atria and regulation of the blood back into the atria during ventricular contraction. The walls of the ventricle are lined with trabecular carnea, ridge of cardiac muscles covered by endocardium. In addition to this muscular ridge, a band of cardiac muscles also covered by endocardium. known as the moderator band, reinforces the thin walls of the right ventricles and plays a critical role in cardiac conduction. It arises from the inferior portion of the intraventricular septum and crosses the inferior space of the right ventricle to connect with an inferior papillary muscle. When the right ventricle contracts, it ejects blood in the pulmonary trunk, which runs into the left and right pulmonary arteries that carry it to each lung. The superior surface of the right ventricle begins to taper as it approaches the pulmonary trunk. At the base of the pulmonary trunk is a pulmonary semilunar valve that prevents backflow from the pulmonary trunk. Left atrium. After exchange of gases in the pulmonary capillaries, blood returns to the left atrium, high in oxygen via one of the four pulmonary veins, while the left atrium does not contain pectineal muscles. It does have an auricle that includes these pectineal ribs. Blood flows nearly continuously from the pulmonary veins back into the atrium, which act as a receiving chamber, and from here, through an opening into the left ventricle, most blood flows passively into the heart while both the atria and ventricles are relaxed, but toward the end of the ventricular relaxation period, the left atrium will contract, pumping blood into the ventricles. This atrial contraction accounts for approximately 20% of ventricular filling. The opening between the left atrium and ventricle is guarded by the mitral valve. Left ventricle. On figure you can see red colors. Recall that also both sides of the heart will pump the same amount of blood. The muscular layer is much thicker in the left ventricle compared to the right. Like the right ventricle, the left also has trabecular cardia, but there is no moderator band. The mitral valve is connected to papillary muscles via chorda tendinea. There are two papillary muscles of the left, the anterior and posterior, as opposed to three on the right. The left ventricle is a major pumping chamber for the systemic circuit. It ejects blood into the aorta through the aortic semilunar valve. This you can see on figure semilunar valve of aorta. Heart valve structure and function. A transverse section through the heart slightly above the level of the atrioventricular septum reveals all four heart valves along the same plane. The valves ensure unidirectional blood flow through the heart. Between the right atrium and the right ventricle is the right atrioventricular valve, or tricuspid valve. It typically consists of three flaps or leaflets made of endocardium reinforced with additional connective tissue. The flaps are connected by chorda tendinea to the papillary muscle, which controls the opening and closing of the valves. Imagine from the right ventricles at the base of the pulmonary trunk is a pulmonary semilunar valve or pulmonary valve. It's also known as the pulmonic valve or the right semilunar valve. The pulmonary valve is comprised of three small flaps of endothelium reinforced with connective tissue. When the ventricle relaxes, the pressure differential causes blood to flow back into the ventricle from the pulmonary trunk. This flow of blood fills the pocket-like flaps of the pulmonary valve, causing the valve to close and producing an audible sound. Unlike the atrioventricular valves, there are no papillary muscles or chorda tendinea associated with the pulmonary valve. Located the opening between the left atrium and left ventricle is a mitral valve, also called like bicuspid valve or the left atrioventricular valve. Structurally, this valve consists of two cusps compared to the three cusps of the tricuspid valve. In a clinical setting, this valve is referred to as the mitral valve rather than the bicuspid valve. The two cusps of the mitral valve are attached by chorda tendinea to papillary muscles that project from the wall of the ventricles. At the base of the aorta is aortic semilunar valve, or the aortic valve, which presents backflow from the aorta. It normally is composed of three flaps. When the ventricle relaxes and blood attempts to flow back into the ventricle from the aorta, blood will fill the cusps of the valve, causing it to close and producing an audible sound. When the ventricle begins to contract, pressure within the ventricles rises and blood flows toward the area of lowest pressure, which is initially in the atrium. This backflow causes the cusps of the tricuspid and mitral bicuspid valves to close. These valves are tied down to the papillary muscles by chorda tendinea. During the relaxation phase of the cardiac cycle, the papillary muscles are also relaxed into the tension on the chorda tendinea is slight. However, as the myocardium of the ventricle contracts, so do the papillary muscles. This creates tension on the chorda tendinea, helping to the atrioventricular valves in place and preventing them from being blown back into the atria. The aortic and pulmonary semilunar valves, like the chorda tendinea and papillary muscles, associated with the atrioventricular valves. Instead, they consist of pocket-like folds of endocardium reinforced with additional connective tissue. When the ventricles relax and the change in pressure forces the blood towards the ventricles, the blood presses against this cusp as seals the opening. Circulation through the heart. On figure you can see circulation scheme of the venous and arterial blood in our body. There are two distinct but linked circuits in the human circulation called the pulmonary and systemic circuits. Although both circuits transport blood and everything it carries, we can initially view the circuit from the point of view of gases. The pulmonary circuit transports blood to and from the lungs, where it picks up oxygen and delivers carbon dioxide for exhalation. The systemic circuit transfers oxygenated blood to virtually end of the tissues of the body and returns relatively deoxygenated blood and carbon dioxide to the heart to be sent back to the pulmonary circulation. The right ventricle pumps deoxygenated blood into the pulmonary trunk, which leads towards the lungs and bifurcates into the left and right pulmonary arteries. These vessels in turn branch many times before reaching the pulmonary capillaries. When gas exchange occurs, carbon dioxide exits the blood and oxygen enters. The pulmonary trunk arteries and they branch are the only arteries in the postnatal body that carry relatively deoxygenated blood. Highly oxygenated blood retaining from the pulmonary capillaries in the lungs passes through a series of vessels that join together to form pulmonary veins, the only postnatal veins in the body that carry highly oxygenated blood. The pulmonary veins conduct blood in the left atrium, which pumps the blood into the left ventricle, which in turn pumps oxygenated blood into the aorta and on the main branch of the systemic circuit. Eventually, these vessels will lead to the systemic capillaries, where exchange with the tissues fluid and cells of the body occur, in this case oxygen and nutrition, exit the systemic capillaries to be used by the cells in the metabolic process, and carbon dioxide and waste product will enter the blood. The blood exciting the systemic capillaries is lower in oxygen concentration than when it enters. The capillaries will ultimately unite to form venules, joining to form ever large veins, eventually flowing into the two major systemic veins, the superior vena cava and inferior vena cava, which return blood to the right atrium. The blood in the superior and inferior vena cava flows into the right atrium, which pumps blood into the right ventricle. This process of blood circulation continues as long as the individual remains alive. Understanding the flow of blood through the pulmonary and systemic circuits is critical to all health professions. Function properties of the heart. The heart has basic properties which are essential for in functioning as the central pump of the cardiovascular system. There are autorhythmicity, conductivity, excitability, refractoriness, and contractility. Automatism, ability of initiate an electrical impulse. Conductibility, ability to transmit an electrical impulse from oneself to another. Excitability, ability to respond to an electrical impulse. Refractoriness, cardiac muscles cannot be excited during the whole periods of systole and early part of diastole. This period prevents wave summation and tetanus. And contractility. Contractility is the ability of the cardiac muscles to contract. In this way, flowing of blood is provided. Electrophysiological properties of myocardial contractile cells. The level of the resting potential in contractile cardiomyocyte is within -90 to -95 millivolts and it's stable. Resting potential of myocardial contractile cells arises due to diffusion of potassium ions from the cells and entrance of chloride ions to cardiomyocytes. But in contrast to the phase cross-line muscles, chronic and chloric permeability of the membrane is more small than potassium and plays a minor role of the formation of the resting potential of contractile cardiomyocyte. The action potential recorded in the ventricular muscles fiber shown in figure average about 105 millivolts, which means that the intercellular potential rises from a very negative value about -85 millivolts between beats to a slightly positive value about +20 millivolts during each beat. After the initial spike, the membranes remain depolarized for about 0.2 seconds, exhibiting a plateau followed at the end to the plateau by abrupt repolarization. Cardiac muscle action potential. Figure summarizes the phases of the action potential in the cardiac muscles and the ion flows that occur during each phase. Phase 0, depolarization. An action potential triggered in the neighboring cardiomyocyte of pacemaker cells causes the transmembrane potential to rise about 90 millivolts. It's caused by a repeat influx of sodium into the cells. Fast ions channels start to open on by one and sodium leaks into the cells, further raising the T transmembrane potential. Transmembrane potential approach -70 millivolts. The threshold potential is cardiomyocyte is the point at which enough fast sodium ions channels have opens to generate a self-sustaining inward sodium currents. The light sodium currents rapidly depolarize the TMP to 0 millivolt and slightly above 0 millivolt for a transient periods of time called the overshoot, fast sodium ion channels close. Recall that fast sodium channels are time dependent. L-type may be long opening calcium ion channels, open when the TMP is greater than minus 40 millivolts and cause a small but steady influx of calcium ions down its concentration gradients. Phase number 1, initial repolarization. Fast sodium channels is closed. The cells begin to repolarize and potassium ions leave the cell through the open potassium channels. Transmembrane potential is now slightly positive. Some potassium channels open briefly and an outward flow of potassium ions retain the transmembrane potential to approximately 0 millivolts. Next phase, phase number 2, plateau. Calcium channels open and fast potassium channel is closed. A brief initial repolarization occurs and the action potentials that plateau as a result of increased calcium ion permeability and decreased potassium ion permeability. The voltage-gated calcium on channels open slowly during phases fast and 0, and calcium enter the cell. Potassium channels then close, and the combination of decreased potassium ion efflux and increased calcium ion influx causes the action potential to plateau. Phases number 3. Repeat repolarization. Calcium channels close and slow potassium channels open. The closure of calcium ion channels and increased potassium ion permeability, permitting potassium ions to rapidly exit the cell, ends the plateau and return the cell membrane potential to its resting level. Calcium ion channels are gradually inactivated. Persistent outflow of potassium ions now exiting calcium inflow bring transmembrane potential back toward resting potential of 90 millivolts to prepare the cells for a new cycle of depolarization. Normal transmembrane ionic conversion gradients are restored by retaining sodium ions and calcium ions to the extracellular environments and potassium ions to the cells inferior. The pumps involved include the sarcolemma sodium-calcium exchanger, calcium ATPase, and sodium-potassium ATPase. Last phase, phase number 4, is resting membrane potential, average about minus 90 millivolts. Electrophysiological properties of atypical cells. In the cells of the conduction system, resting potential is absent. Instead, present phase of slow spontaneous diastolic depolarization, which is due to enter through the membrane of cardiomyocyte calcium ions, leading to accumulation in the cells of passively charged ions. Upon reaching by membrane potential critical points of the depolarization about minus 60 millivolt action potential spontaneously arises and spread by conduction system and tends to the contractile myocardium. The SA node is a normal pacemaker of the heart. The configuration and ionic basis for its action potential differ in several important respects from those ventricular fibers. The following features of the action potential of the SA node are different from those in ventricles. The SA node exhibits automaticity, that it can spontaneously generate action potential without neural input. It has an unstable resting membrane potential, indirect, constant to cells in ventricular. It has no sustained plateau. The phases of the SA node action potential are described here and correspond to the number phases shown in figure. Phase 0, upstroke. Phase 0 is the upstroke of the action potential. Note that the upstroke is not the repeat or the sleep as in the other types of cardiac tissues. In the other myocardial cells, the upstroke is a result of increase in gate sodium and an inward sodium ions current. In the SA nodal cells, the upstroke is a result of increase in gate calcium and an inward calcium ion current carried primarily by L-type calcium ion channels. There are also T-type calcium ion channels in SA node, which carry part of the inward calcium ion current in the upstroke. Phases first and second are absent. Phase 3, repolarization. As in the other myocardial tissues, repolarization in the SA node is due to an increase in concentration potassium ion. Because its electrochemical driving forces on potassium ion are large, both chemical and electrical driving forces favor potassium ion leaving the cell. There are an outward potassium ion current which repolarized the membrane potential. And phases number 4, spontaneous depolarization or pacemaker potential. Phase 4 is the longest portion of the SA node action potential. This phase accounts for the automaticity of SA nodal cells, the ability to spontaneously generate action potential without neural input. During phase number 4, the most negative value of the membrane potential, called the maximum diastolic potential, is approximately minus 65 millivolt, but the membrane potential does not. The conducting system of the heart. Components of the cardiac conduction system involve the spread of electrical activity from the sinoatrial node to the atrioventricular node, down the bundle of His, and along the Purkinje fibers, as the electrical activity spread along the heart's conduction system initiates myocardial contraction in the surrounding myocardial tissue. Sinoatrial node. The sinoatrial node is a group of cells found high up in the right atrium close to its junction with the superior vena cava. The sinoatrial node functions as the heart's intrinsic pacemaker regulating heart rate. The sinoatrial node spontaneously generates electrical impulses which are transmitted to the right and left atrium. These electrical impulses stimulate the atrial myocardium to contract. Atrial muscles conduct relatively fast 0.5 meter per second. Atrioventricular node. The atrioventricular node is a group of specialized cells situated in the atrioventricular septum, just above the coronary sinus ostium, that receives electrical impulses from the atria and that transmit the electrical impulse from the atria to the ventricles. The AV nodes have a slower conduction velocity, near 0.05 meter per second, than the atria, allowing maximal ventricular filling prior to contraction. The bundle of His is a collection of heart muscle cells specialized for electrical conduction. These cells receive input from the AV nodes. The bundle of His branch into the left and right bundle, branch which travel down the interventricular septum. Branch of the bundle of the His propagate impulses to the left and right ventricles, respectively. Each branch terminates in several Purkinje fibers. Purkinje fibers are situated in the subendocardium. They transmit the wave of electricity to the ventricle myocardium. This wave of electricity results in ventricular contraction. Autorhythmicity. Autorhythmicity is a myogenic, innate property of the heart independent of its nerve supply. The conducting system of the heart is responsible for generating rhythmic, electrical impulses which drive the heart to beat in a rhythmic way. The focus which generates the highest frequency of impulses is one that drives the whole heart and called the pacemaker of the heart. The normal pacemaker of the heart human is SA node, sinoatrial node. There are several possible sites in the conducting system that can act as possible pacemaker. Sinoatrial SA node, atrioventricular AV node, and Purkinje system. In the primary pacemaker is damaged or blocked. The secondary pacemaker take over. If the flow of impulses from the atria to the ventricles stop completely, the tertiary pacemaker takes over and the ventricles continue to beat at the idioventricular rhythm. The pacemaker cells are characterized by having an unstable membrane potential. After firing in action potential, the membrane potential decreases. The membrane potential depolarizes gradually from a basal value of -60 mV to a critical firing level of -45 mV. At this level, an action potential is fired and the cycle is repeated. The gradual depolarization of the SA node cells is called the pacemaker potential or the pre-potential. The early part of the pacemaker potential is caused by a decrease in the permeability of the membrane to potassium ion. The late part is caused by calcium ion influx through the transient T-type calcium channels. Automatism to the heart. The ability of cells of the heart conducting system to produce independently bioelectrical impulses that causes its excitement. Structure of conducting system have different degrees of automaticity. It establishes the so-called gradient of automaticity. It manifests itself in a reduced ability to automatism of different structures of the conducting system according to their distance from the sinoatrial node. Thus, in the sinoatrial node, number of action potential reach the level of 60 to 90 impulses per minute, and in the cells of his node from 30 to 40 impulses per minute, and fibers of Purkinje less than 20 impulses per minute. Gradient of automaticity caused by different spontaneous permeability cells membrane of conduction system to calcium ions. Based on the fact the sinoatrial node impulse its rhythm to the departments of conduction system that lying lower, it's called pacemaker or pacemakers of first order. Pacemaker of second order is atrioventricular node. Pacemaker of third order is his bundle, his node, and its ramifications. The assimilation of rate. Under normal circumstances, the automaticity of all section of the conduction system is suppressed by sinoatrial node, which enforces its own rhythm. That is why all parts of the conduction system begin to work at the same pace, also they have their own rhythm. The phenomenon in which the structure with slow pace of generating of action potentials assimilate more frequent rhythm from other parts of the conducting system called the assimilation of rate. Assimilation of rate. Transmission of excitation in the atria and AV node. Transmission of excitation in the atria. The excitement that arose in sinoatrial node is conducted through the atria at a spread and speed of 0.8 to 1 meter per second. All fast depolarization covers the right atrium and then left atrium. Time of coverage by excitation of both atria equals 0.1 seconds. Conduction of excitation in the atrioventricular node. With the transfer of excitation from the atria to the ventricles, its delay in atrioventricular node is observed. It is associated with features of geometrical structure of node and the specific development electrical potential in it. This is due to the low density of sodium ion channels. This delay is important for sequential excitation and contraction of atria and then the ventricles. The speed of excitation spread through the atrioventricular node is about 0.02 meter per second. Figure shows transmission of the cardiac impulses through the atria in our heart. Transmission in the ventricular Purkinje system. Transmission of the excitation through the His bundle and the Purkinje fibers is 1.5 meter per second. The process of ventricular depolarization begins at the middle third of the interventricular septum and extend to the top and side wall of the right and left ventricle, basal part of the ventricles, and the upper third of the intraventricular septum are depolarized at last. Next delay of excitation in the place of contact of Purkinje fibers with contractile myocytes. It is the result of summation of action potential which contribute to the synchronization of myocardial excitation, the speed of excitation conduction within ventricles average 0.3 to 0.9 meter per second. Transmission in the ventricular Purkinje system you can see on this figure. On scheme, you can see conduction of excitation in the heart. First step, SA node, activity and atrial activation begin. Time 0. Next step, step number 2, stimulus spread across the atrial surface and reach the AV node. Elapsed time equal 50 milliseconds. Step number 3. There is a 100 milliseconds delay at the AV node. Atrial contraction begins. Elapsed time equal 150 milliseconds. Next step, step number 4. The impulse travels along the interventricular septum with the AV bundle and the bundle branch to the Purkinje fibers and via the moderator band to the papillary muscles of the right ventricle. Elapsed time equal 175 milliseconds. Step number 5. The impulse is distributed by Purkinje fibers and relates throughout the ventricular myocardium. Atrial contraction is completed and ventricular contraction begins. Elapsed time equal 225 milliseconds. The students' scheme show about condition of excitation in the heart. Excitability. Excitability is the ability to respond to stimuli. The resting membrane potential of the contractile fibers is stable at about minus 90 millivolts. When an effective stimulus is applied, a propagate action potential is produced in the cell membrane. Excitability is the capacity of myocardial cells To generate action potential is response to inward depolarizing current. Straightly speaking, excitability is the amount of inward current required to bring a myocardial cells to the threshold potential. The excitability of a myocardial cells varies over the course of the action potential, and this change in excitability are reflected in the refractory periods. Absolute refractory period. For most of duration of the action potential, the ventricular cell is completely refractory to fire another action potential. No matter how large a stimulus might be applied, the cell is incapable of the activation gates open. Therefore, during those phases of the action potential, when the membrane potential is depolarized, a portion of the sodium ion channels will be closed because the inactivation gates are closed. When the sodium ion channels are closed, inward generating a second action potential during the absolute refractory periods. Because most of sodium ion channels are closed, the absolute refractory periods include the upstroke, the entire plateau, and a portion of the repolarization. This period concludes when the cells has repolarized to approximately minus 50 millivolts. Depolarizing current cannot flow through them, and there can be no upstroke. Without an upstroke, a normal action potential cannot occur and the cells in refractory. Once repolarization occurs, the inactivation gates of the sodium channels open and the cells in once again excitable. Effective refractory periods. The effective refractory periods includes and is slightly longer than the absolute refractory periods. At the end of the effective refractory periods, the sodium channels start to recover. The distinction between the absolute and effective refractory periods is that absolute means absolutely no stimulus is large enough to generate another action potential. Effective means that a conducted action potential cannot be generated. Relative refractory period. The relative refractory periods begin at the end of the absolute refractory periods and continues until the cell's membrane has almost fully repolarized. During the relative refractory periods, even more sodium ion channels have recovered, and it's possible to generate a second action potential. Also, the greater than normal stimulus is required. If a second action potential is generated during the relative refractory period, its action potential is generated during the relative refractory period, it will have an abnormal configuration and a shortened plateau phase. Supranormal periods. The supranormal periods follow the relative refractory periods. It begins when the membrane potential is minus 70 millivolts and continues until the membrane is fully repolarized back to minus 85 millivolts. As the name suggests, and cell is more excitable than normal during the period. In other words, less inward current is required to depolarize the cells of the threshold potential. The physiological explanation for this increased excitability is that the sodium channels are recovered, and because the membrane potential is closer to threshold, that is the rest in either of fire and action potential than when the cell's membrane is the resting membrane potential. It lasts for the warm periods of systole and the early part of diastole. Contractility. Contractility is the ability of the muscles to convert the potential energy of food into mechanical energy. Physiologic anatomy of cardiac muscles. Figures show the histology of cardiac muscles demonstrating cardiac muscle fibers arranged in the left side work, with the fibers dividing, recombining, and then spreading again. Cardiac muscles is striated in the same manner as in skeletal muscles. Cardiac muscles has typical myofibril that contain actin and myosin filaments, almost identical to those found in skeletal muscles. These filaments lie side by side and slide during contraction in the same manner as occur in skeletal muscles in other ways. However, cardiac muscles in quite different from skeletal muscle. Cardiac muscles in the syncytium. The dark areas crossing the cardiac muscle fibers are called intercalated discs. They are actually cell membranes that separate individual cardiac muscle cells from one another. That is, cardiac muscle fibers are made up of many individual cells connected in series and in parallel with one another. At each intercalated disc, the cell membranes fuse with one another to form permeable communicating junction that allow rapid diffusion of ions. Therefore, a functional point of view, ions move with ease in the intercellular fluid along the longitudinal axis of the cardiac muscles fibers, so that action potential travel easily from one cardiac muscle cell to the next, past the intercalated disc. Mechanism of myocardial contraction. Under the influence of the action potential, calcium ions from extracellular space and from the cisterns of the sarcoplasmic reticulum entering the myoplasma, where under its influence protein troponin, which push back tropomyosin from actin active sites, conforms. Therefore, between actin and myosin, bridges are formed. Thus, splitting of ATP takes place in these periods, and its energy is used for sliding of actin filaments. The more calcium ions from the troponin contained, the more actomyosin bridge formed, and the greater the force of muscle contraction. Excitation-contraction coupling. As in skeletal and smooth muscles, Excitation-contraction coupling in cardiac muscles translate the action potential into the production of tension. The following steps are involved in excitation-contraction coupling in cardiac muscles. These steps correlate with the cycled numbers shown in figure. The cardiac action, number 1. Cardiac action potential is initiated in the myocardial cells membrane as the depolarization spread to the interior of the cells via the T-tubules. Recall that a unique feature of the cardiac action potential is plateau, which results from an increase in gate calcium and an inward calcium ion currents in which calcium ion flows through the L-type calcium channel from extracellular fluid to intracellular fluid. Second, entry of calcium ion into the myocardial cells produce an increase in intracellular calcium concentration. This increase in intracellular calcium concentration ions is not sufficient alone to initiate contraction, but it triggers the release of more calcium ion from stores in the sarcoplasmic reticulum through calcium release channels. This process is called calcium-induced calcium release, and these calcium ions that enters during the plateau of the action potential is called the trigger calcium. Two factors determine how much calcium ions is released from the sarcoplasmic reticulum in this step. The amount of calcium ions previously stored and the size of the inward calcium ion current during the plateau of the action potential. Number 3 and number 4. Calcium ion released from the sarcoplasmic reticulum causes the intercellular calcium ions concentration to increase even further. Calcium ions now binds to troponin C. Tropomyosin is moved out of the way, and the interaction of actin and myosin can occur. Actin and myosin bind cross-bridge, form, and the break, and tin and thick filaments move past each other, and tension is produced. Cross-bridge cycling continues as long as intracellular calcium ions concentration is high enough to occupy to the calcium ions binding site of troponin C. Number 5. A critically important concept is that the magnitude of the tension developed by myocardial cells is proportional of the intracellular calcium ion concentration. Therefore, it's reasonable that hormones, neurotransmitters, and drugs that after the inward calcium ion current during the action potential plateau, or that along sarcoplasmic reticulum calcium ion stores would be expected to change the amount of tension produced by myocardial cells. Factors that affect cardiac contractility. Mechanical factors are preload and afterload, cardiac factors, and extracardiac factors. The preload. The preload is a load that determines the initial length of the resting muscle before contraction. The level of the preload is represented by the end diastolic volume. By the venous return, it affects the tension developed in the muscles. When the venous return increases, the strength of ventricular contraction increases too, leading to an increase in the stroke volume. This Frank-Starling law. Afterload is a load the muscle faces when it begins to contract. In this interact heart afterload is produced by the aortic impedance, which is determined by aortic pressure, arterial systolic blood pressure, arterial wall rigidity, and blood viscosity. Cardiac factors. The myocardial mass. The myocardial mass, a significant injury or loss of the functioning ventricular muscle, decrease the force of myocardial contractility. This also occurs in cases of heart failure. The heart rate. The force of cardiac contractility is affected by the frequency of stimulation. An increase in the frequency of stimulation causes a proportional increase in the force of contraction. Extracardiac factors. There are neural, physical, and chemical factors. Neural factors. This influence sympathetic and parasympathetic part of autonomic nervous system. Sympathetic stimulation and norepinephrine exert a positive inotropic effect by increasing cyclic EMP in cardiac muscle fibers and activation of calcium ion channels and increased calcium influx. Conversely, parasympathetic stimulation and acetylcholine exert a negative inotropic effects, but only the atrial muscles, since the vagus nerves don't supply the ventricles. Psychical factor, physical factors. A moderate rise of the body temperature strengthens cardiac contractility. The calcium ion influx and ATP formation in the muscles, while the excessive rise of the body temperature exhausts the metabolic substances in the cardiac muscles and decreases its contractility. Hypothermia also decreases cardiac contractility. Chemical factors. Different hormones, catecholamines, epinephrine and norepinephrine, and dopamines, glucagon and thyroid hormones all exert a positive inotropic effects. Blood gases, moderate hypoxia and hypocapnia increase the cardiac contractility, whereas severe hypoxia and hypocapnia directly depress the cardiac muscles and decrease its contractility. Hydrogen ions concentration, an increase on blood, drop of the blood produce a negative inotropic effects, whereas a decrease of the blood and rise of the blood pH, this alkalosis produce a positive inotropic effects. Next, information about mechanism of relaxation of the heart muscles. The relaxation of cardiomyocytes occurs as a result of repolarization of the membrane is based on the fact is the impact of repolarization removes calcium ions from the contractile proteins. After that, calcium captured by pumps of synthesis of sarcoplasmic reticulum. It also displayed in the interstitial liquid due to the pump work of cell's membrane. The main process that determines the relaxation of cardiomyocytes, the removal of calcium ions from sarcoplasma, resulting in decreasing of calcium ion concentration. Thus, complexes of calcium ion with troponin C break tropomyosin, sheds according to actin filaments, and closes the active center's contraction is terminate. All information you can see on this slide. Relaxation. Mechanism of relaxation is calcium is cleared from the sarcoplasm by the sodium-potassium exchanger and two calcium pump. One in the sarcoplasmic reticulum that reuptakes calcium back into the reticulum and another in the sarcolemma that pumps calcium out into the extracellular fluid. The clearance of calcium from the sarcoplasm terminates the contraction and stands the relaxation process. Last information about difference between skeletal and cardiac muscles. Look please on table. First time information about membrane potential. For skeletal muscles, membrane potential equal near minus 70. For contractile myocardium, minus 90. Next information about events leading to threshold potential. For skeletal muscles, sodium ions enter through the acetylcholine operated channels, and for cardiac muscles, depolarization enters through the gap junction. Next information about rising phase of action potential. For skeletal muscles is sodium entry, and for contractile myocardium the same sodium entry. Next information about repolarization phase. For skeletal muscles, caused by potassium efflux. For cardiac muscles, extended plateau and movement calcium ions and potassium ions. This last information about differences between skeletal muscles and cardiac muscles and action potential. Thank you for your attention.